All right, welcome to The Explainer. You know, your immune system is just this incredible masterpiece of evolution, a complex army that's defending you every single second. But what happens when parts of that army fail? So today we're going to flip the script. We're going to explore the immune system not through its wins, but through its super rare and honestly super revealing failures.
Okay, so just think about this for a second. What if one of your body's most critical first responders gets the 911 call, right? An infection is happening, but they're just stuck. They literally can't leave the station to get to the scene. What do you think happens then?
So that first responder we're talking about, that's the phagocyte. It's a type of white blood cell. You've probably heard of neutrophils, that's one of them. And you really can think of it like an elite soldier. Its whole job, its entire mission, is pretty straightforward but absolutely critical. Hunt down any bad guys. We're talking bacteria, fungi, gobble them up, and just completely obliterate them.
Right, but here's the catch. For our little soldier to actually do its job, it has to get to the battlefield in the first place. See, most of the time, these cells are just cruising through your bloodstream, kind of like a police patrol. But when an alarm bell goes off, a signal that there's an infection in, say, your skin, they have to pull off this incredible, almost acrobatic maneuver to get out of the blood vessel and into the action.
So here's how it's supposed to go down, in 3 steps. First, the cell starts to slow down, kind of tumbling and rolling along the inner wall of the blood vessel, like a ball on a sticky floor. Then, step 2, it just slams on the brakes, hard. It grabs onto that wall in a step we call tight adhesion. And finally, it does this amazing contortionist act where it squeezes itself between the cells that make up the vessel wall and pops out on the other side, right into the infected tissue. So it's roll, stop, squeeze. Simple as that.
Now that braking system, that's where things get really fascinating. To stop, the phagocyte uses this special protein called CD18, and you can seriously just picture it like molecular Velcro. It lets the cell get a super tight grip on the vessel wall. Well, in this incredibly rare genetic condition called leukocyte adhesion deficiency, LAD1 for short, that CD18 Velcro is just missing. It's defective. So the cell can do the first part, it can roll along, but it can never, ever stop. It just gets swept away by the bloodstream, completely unable to get to the infection.
And what's wild is that this one tiny molecular problem leads to some really specific telltale signs. Patients get these terrible recurring bacterial infections. And if you do a blood test, you'll see a sky-high white blood cell count. Why? Well, because all the soldiers are in the blood, they're just stuck in traffic. They can't get out. But here's the absolute kicker, the thing you have to remember. Their infections don't form pus. Think about it. What is pus? It's mostly just a pile of dead neutrophils that showed up, fought the battle, and died. If your soldiers can never arrive at the battle, you're not going to have a pile of their dead bodies, right? No pus.
Okay, so that's problem number 1. The soldiers can't even get to the fight. But what if they do? What if they get there perfectly? They surround the enemy, but then their main weapon is just broken. It won't fire. That brings us to our next major glitch in the system.
So this primary weapon the phagocyte has is a chemical bomb, basically. It's called the respiratory burst. And no, it has nothing to do with breathing. It's all about the cell rapidly creating this toxic cloud of oxygen molecules. We're talking things like superoxide, hydrogen peroxide, to literally blast the germs it's eaten into oblivion. And the whole thing is powered by this really powerful enzyme engine called NADPH oxidase. Now, in a condition called chronic granulomatosis disease, or CGD, that engine, that NADPH oxidase enzyme, is broken. So picture this. The soldier gets to the battlefield, it successfully swallows the bacterium, and then crickets. Nothing. The respiratory burst just fails to fire. The cells simply can't make the toxic chemicals it needs to actually kill the invader it's trapped inside.
This specific defect creates a really specific vulnerability. It means patients are in big trouble from a particular list of germs we call catalase positive, things like Staph aureus and Aspergillus. These bad guys are clever. They produce their own enzyme, catalase, which is like a shield. It neutralizes the little bit of backup peroxide the cell might try to use. So you've got a soldier with a broken primary weapon, and the enemy has a perfect defense against its weak little backup plan. It's a disaster. And this is where the diagnostics get really cool, because we can actually test this weapon system directly. There's an older test, the NBT test, where you use a special dye. If the enzyme is working, the dye turns blue. In CGD, nothing happens. It just stays yellow. The newer test, the DHR test, is even more slick. It uses a machine called a flow cytometer. A healthy cell will light up, showing this big shift in fluorescence when it makes the burst. But a CGD cell, the line stays flat. No shift at all.
We are literally watching the weapon fail to fire in real time. It's incredible.
Okay, but it's not always the frontline soldier's fault. Sometimes the problems are deeper inside the cell, with the internal logistics, the supply lines. Or sometimes the problem is with the air support that's supposed to be backing them up. So let's look at two more examples. On the one hand, you have Chidiac-Higashi syndrome. Here, the soldier swallows the germ, but the internal garbage disposal system is broken. The little bubble with the germ inside can't fuse with the bubble that has all the digestive enzymes. This leads to these huge, totally useless clumps inside the cell, and for reasons that are super complex, it also causes partial albinism. Then you have myeloperoxidase deficiency. This one's usually much milder. It's just the very last step in the chemical weapon assembly line that's missing, turning hydrogen peroxide into what's basically bleach. For most bacteria, the body has other ways to cope, but it can lead to stubborn infections with fungi, like Candida.
So moving beyond just the single cell, there's this whole other part of your initial defense system called the complement system. And the best way to think about this is like air support for your ground troops. It's a whole cascade of over 30 different proteins just floating in your blood, ready to go. They can either kill pathogens directly, or, just as importantly, they can paint a target on them, making them way easier for our phagocyte soldiers to find and destroy.
And here is what is so elegant and so fascinating about this system. Depending on where in that protein cascade the failure happens, you get completely different diseases. So, if the early proteins in the chain are broken, your body can't clean up cellular waste properly, and that can lead to autoimmune diseases like lupus. If you're missing the absolute key central protein, C3, then you can't paint the target on bacteria, and you get really severe, overwhelming infections. And get this, if the late components are missing, you can't build this drill-like weapon called the membrane attack complex, and that creates a very, very specific weakness to one particular type of bacteria, Neisseria. It just goes to show that every single piece of the chain has a highly specialized job.
Okay, so we've just gone through all these really specific, really rare ways the whole system can fall apart. So what's the point? Why should we care so much about studying these, what some people call experiments of nature?
It matters because every single one of these failures teaches us something absolutely fundamental about how the system is supposed to work. LAD, the disease where the cells can't stick, proves that CD18 Velcro is non-negotiable. You have to have it to get cells to an infection. Period. CGD, where the weapon doesn't fire, proves the absolute critical role of that NAPDH oxidized engine for killing a very specific list of bad guys. And the complement deficiencies, they show us that different parts of the same system are hyper-specialized to deal with different kinds of threats. It's beautiful, really. So here's the big takeaway, the whole point of this. It's by studying what breaks that we learn how the machine is supposed to work in the first place. These rare glitches, these single-gene screw-ups, they give us the clearest, most undeniable blueprint for our body's entire defense network.
You know, it's really a stark reminder that our immune system is constantly walking this incredible tightrope between defending us from invaders and accidentally attacking ourselves. For every unbelievably powerful weapon it has, there has to be a control system, a safety switch. For every soldier it sends into battle, there has to be a clear mission and a way to call them back. And it's by looking at these rare moments where that balance is lost, where the system glitches, that we get the absolute clearest picture of how miraculously it keeps us safe every other day of our lives.